Hey everyone. This chapter moves us in a new direction, so I'm kind of excited about tackling a new topic. I'm going to be talking about memory modulation systems. In other words, rather than talking about the direct neuronal mechanisms that underlie synaptic plasticity, I'll be describing systems that modulate the preparedness of neurons to engage in learning and memory, including the role of the amygdala in recognizing emotional salience and the roles of the hypothalamus and pituitary in activating hormonal responses that modulate the learning experience. Even the liver will be shown to play a role. Whoever thought they would come into a psychology course and talk about the liver? Go figure. Anyway, we have a lot to cover, so let's do it. In most of the information I've provided throughout this course so far, I've focused on specific cellular and biochemical mechanisms that underlie learning and memory. We've spent most of the semester inside the postsynaptic spine. Okay, not literally, but you know what I mean. Well, we will now, figuratively step out of the spine for a little bit and examine the multiple independent systems that can converge to impact memory storage mechanisms. These multiple independent systems, these memory modulators, are not actually part of the storage mechanisms themselves. They're not part of the biochemical mechanisms inside the spines, and they only work in the short term. In other words, in the memory modulating systems, there's no equivalent to PKM zeta. There's no enduring activity. Rather, they activate in response to stimulus situations at the moment, and they provide their contribution to modulate the memory activating and consolidating processes, and then they shut down when the situation is resolved. Now, one more important introductory point I would like to make about memory modulators is that they're not required for retrieval. The storage of the memory may be influenced by the modulators at the time of the encoding, but it's just not important at the time of retrieval. Now you may be thinking, that's obvious, isn't it? Why would an activation of some emotional or hormonal system that is active at the time of the learning of the memory be involved in memory retrieval? Well, this is an issue that many in the public media have hyped over the years. The notion of state-dependent learning suggests that if you are perhaps hyper-aroused at the time of the learning, you will remember the material better if you're hyper-aroused at the time of recall. Or if you're perhaps a little bit drunk at the time of learning, then you will remember the material a little bit better the next time you're a little bit drunk. If that sounds a little bit silly to you, well, I agree. There have been various reports of state-dependent learning, even in the scientific literature over the years. But each time one of these reports surfaces, it turns out that there's some experimental bias or flaw driving the outcome. Let me make this clear. There is no scientifically credible evidence for state-dependent learning. Memory modulators can influence your acquisition and storage of a memory, but you do not need to return to the state that you were in during acquisition in order to recall the memory. Now, all of that is not to say that these same systems that modulate memory acquisition and consolidation can't influence retrieval. Certainly, if you're hyper-aroused, it may actually be challenging to focus and engage in an effortful recollection of an event. The point is that there is no association between the state conditions like arousal, or drunkenness for that matter, at the time of acquisition, and the state conditions required for retrieval of the memory. Okay, on this slide, I've actually modified the diagram from page 242 of your textbook to show you that activation of the hypothalamic pituitary adrenal axis makes an important contribution to memory modulation, and also to indicate that adrenergic innervation extends all over the brain, not just to the amygdala. as is depicted in the diagram in your textbook. This is still a really silly oversimplification of complex circuits in the brain and modulatory hormonal inputs, but it will do to get us started, and I hope it puts into focus at least some of the systems that we will discuss today. The kinds of synaptic mechanisms we've been talking about are now indicated with the red arrows, where learning experiences directly impact neurons of the amygdala, cortex, hippocampus, caudate, cerebellum, and other brain regions. The blue arrows indicate the memory modulators. You can see that activation of the HPA axis results in systemic release of epinephrine from the adrenal medulla. Following the blue arrows and neurons illustrates that the circulating epinephrine activates brainstem noradrenergic systems that impact the amygdala and other brain regions. The blue arrows also illustrate that activation of the HPA axis causes release of the glucocorticoid hormone cortisol, or, well, corticosterone, depending on if you are a human or a rat, to influence the amygdala and other brain regions. Clearly, the amygdala is implicated both in direct learning of memory, as we've seen, and in the memory modulating actions that we will be discussing in this lecture. So let's take a closer look at the amygdala. The figure at the right is a Nissl-stained section of rat brain cut through the amygdala in the right hemisphere. I've drawn a red box around the area where the blown-up image of the Nissl-stained section originates. The Nissl stain, of course, most clearly labels all cell bodies in the neurons. And if you look closely, you can kind of see the cellular architecture that delineates the subnuclei of the amygdala. So conveniently, those are kind of outlined for you with dotted red lines. But to aid you further in identifying the structures of the amygdala, I included a diagram showing the locations of the lateral and basolateral amygdaloid nuclei, as well as the central nucleus of the amygdala. The basolateral amygdala is a site of convergence of a great many sensory inputs to the amygdala, representing all sense modalities and even higher level integrated perceptual information from all over the cortex, as well as inputs from the thalamus, hippocampus, and other subcortical domains. The schematic at the left of the slide shows that the basolateral amygdala processes these inputs and then innervates broad areas of cortex and a variety of subcortical structures. These include hippocampus, caudate, nucleus accumbens, and other areas. Another very important output of the basolateral amygdala is the central nucleus of the amygdala, which receives input from all amygdaloid subregions and processes those convergent inputs to also output to a vast array of cortical and subcortical structures. Our main understanding of the amygdala's function, of course, is that it uses all of this convergent information and processing power to compare incoming information with a lifetime of experience to identify what stimuli pose a threat, and then to activate the appropriate responses to that threat. And those responses may be activation of behavioral fight or flight reactions, regulation of the autonomic nervous system, especially activation of the sympathetic branch, or hormonal responses from autonomic innervation of the adrenal medulla and the hypothalamic pituitary adrenal axis, as well as mobilization of glucose release from the liver and glucose metabolism within the muscles and so on. In fact, the amygdala is particularly well equipped to engage these responses rapidly, sometimes even before conscious awareness of the threat. In this illustration, you can see that the brain perceives a threat processed by the visual system. Of course, the primary visual system is responsible for processing the perceptual recognition of the stimulus through the thalamus and on through projections to the primary visual cortex and out into the dorsal and ventral streams of visual processing. From the primary visual cortex and higher levels of visual processing, there are also projections that extend to the hippocampus and amygdala. Of course, these are important in cognitive processing of the visual stimulus and for memory incorporation of the stimulus situation, and they are known as the high road of visual processing. However, I want you to notice that there's also a projection that goes directly from the lateral geniculate nucleus of the thalamus right down here to the lateral and basolateral nuclei of the amygdala. This direct projection bypasses the conscious experience of the sensory system and shortcuts directly to the amygdala. So this is known as the low road. Of course, this doesn't mean that the subject doesn't still process the full conscious experience of the situation, but the shortcut, the low road, can mount an immediate fear response that may actually be extremely important from a survival perspective. The graphic on the right side of the slide depicts the organization of the high road and the low road. I should note that there's a high road and a low road for every type of sensory information. Both inputs arrive at the basal and basolateral nuclei and innervate the central nucleus and many other brain regions. The eventual outputs of amygdaloid processing then can regulate behavioral, autonomic, and hormonal responses, as I've already described. One of the earliest observations about the neurochemical basis of learning and memory arose out of the finding that amphetamine at low doses can enhance cognitive performance in a variety of behavioral tests. We all know that the doses are not stoner doses and are well below the doses that students sometimes use to try to up their grades a little. But the finding that low-dose amphetamine can enhance learning and memory implicates noradrenergic neurotransmission, as amphetamine is an indirect noradrenergic agonist. It increases extracellular concentrations of norepinephrine by reversing the norepinephrine transporter. So since the earliest days of investigations into the neurobiology of learning and memory, tons of research has been focused on the role of norepinephrine in learning and memory. Indeed, in the diagram back on slide 2 of this lecture, you can see that adrenergic innervation of many brain areas modulate memory function. So one question was whether adrenergic innervation at all the sites where it inputs has equivalent effects on learning and memory, or if adrenergic innervation at the different sites played different roles in memory acquisition. In 1998, Mark Packard and Lisa Tether tested this by injecting amphetamine into the hippocampus, caudate, or amygdala immediately after training in the Morris Water Maze. Of course, everyone's thinking about what I previously told you about experimental design and is recognizing that the amphetamine injections post-training would be appropriate to avoid any impact on the experience of training per se, and would rather address the impact of adrenergic innervation on consolidation of the learning. And so the animals received one training session consisting of 8 swims, followed by an intracranial injection of a low dose of amphetamine into one of the target areas. When they tested the impact of low-dose amphetamine in the standard Morris-Water maze with the typical hidden platform in probe trials the next day, they found that amphetamine into the hippocampus immediately after training improved learning, as the rats took a shorter period of time to find the location of the platform. In comparison with the performance of rats that were either injected with saline or were injected with amphetamine after a 2-hour delay. The rats injected with amphetamine into the amygdala also benefited from the cognitive enhancing effects of the amphetamine in this test. And in contrast, the rats injected with amphetamine into the caudate nucleus did not benefit from the amphetamine, even when higher doses were used. Then, when Packard and Tether assessed the impact of the amphetamine injections in the visible platform version of the Morris-Water maze, they found that amphetamine injections into the hippocampus had no effect. However, amphetamine into the amygdala increased the learning, or enhanced the learning, as evidenced by the decreased latency to find the platform in comparison with the saline vehicle and delayed amphetamine-treated animals. And amphetamine injections into the caudate also enhanced performance in the visible platform test in comparison with the controls. So it looks like they uncovered some important information about the memory modulating functions of adrenergic inputs to each of these structures. What they identified is that enhanced adrenergic inputs to the hippocampus can evoke a greater learning in the spatial version of the Morris-Water maze. And of course, this seems reasonable when you consider that the hippocampus is so important in spatial learning and memory. Of course, injections of amphetamine into the hippocampus did not impact learning in the visible platform version of the test, because that task is not spatial. It's just a skill or a habit of swimming to the visible platform. In contrast, enhancement of adrenergic innervation of the caudate by intracaudate injection of amphetamine didn't have a positive modulating effect in the spatial hidden platform version of the water maze. But these injections did enhance the outcomes in the visible platform version of the test. This also makes sense when you consider that the caudate does not seem to contribute to spatial learning and memory, but is actually implicated in learning of habits and skills, as we will see further when we get to chapter 18. And then enhancement of adrenergic innervation of the amygdala worked in both versions of the test. I think this also makes sense when you consider that the amygdala is implicated in the emotional content of the swimming challenge. Rats don't like to swim, and so it seems that enhanced modulation of the emotional content of the challenge would promote learning. Of course, within reason, as long as the emotional content doesn't overwhelm the animal and induce a dysfunctional fear state. Overall, the role of the amygdala seemed to be much broader. It seemed to be modulatory in terms of multiple functions, whereas the role of the hippocampus was limited to the spatial component, and the role of the caudate was limited to the skills and habits. Taken one step further, it seems that the amygdala also functions as a modulator of learning and memory functions by influencing both the hippocampal spatial learning system and the striatal skills and habits learning system. Since those experiments there's additional evidence specifically implicating the basolateral amygdala in these functions, and we're going to see some of those experiments coming up shortly. So on this version of the modulator diagram from slide 2, I've labeled the inputs to the basolateral amygdala, where the adrenal medulla releases norepinephrine into the general circulation when the medulla is acted upon by sympathetic innervation. Then that circulating epinephrine binds to receptors on the vagus nerve, those vagus nerve afferents that go to the brainstem. And then in the brainstem, the vagus nerve synapses on neurons of the nucleus of the solitary tract. The solitary tract neurons then project to the locus coeruleus, the major source of adrenergic innervation that extends axons really all over the brain. And of course, one of those targets is the basolateral amygdala, providing it with feedback from the body to inform it about the state of physiological responses to a challenge or a stressor, and situating the basolateral amygdala nicely to modulate additional systems in the brain, as we saw in the experiment that I just showed you. And there is lots of evidence that circulating epinephrine serves as a memory modulator. In 1975, Gold and Buskirk trained rats in an inhibitory avoidance test, and then immediately, or after 1 hour, administered a systemic injection of epinephrine. The rats that were injected immediately with low doses of epinephrine exhibited enhanced learning in the inhibitory avoidance test. Now, higher doses didn't work, and administration of epinephrine at a reasonable low dose after a 1 hour delay didn't work. Note that in contrast to the previous experiments, these are systemic injections of epinephrine rather than intracranial injections of amphetamine. So here, we're implicating the systemic response of the autonomic nervous system and downstream hormones as memory modulators. And there's evidence for the neurocircuitry that I showed you 2 slides previously. King and Williams conducted a cued fear conditioning experiment with a very mild foot shock, hence causing only small increases in freezing in the control animals. They found that if you conduct this experiment in a novel context, the rats learn the association better than they do if you pre-expose them to the environment in which the paired cue and foot shock will be administered. They also found that sotalol, a beta blocker, or a drug that blocks beta adrenergic receptors, blocks the effect of novelty, but does nothing to learning under basal conditions with pre-exposure to the environment. And you can see the diagram at the right shows kind of the same neural circuitry I showed you previously, and they illustrate that the epinephrine binding to adrenergic receptors on the vagus nerve mediates the learning enhancement that occurs when the rats are stressed or autonomically aroused by the novel environment. Now, it's also interesting to note in the lower graph that the effects were equivalent to the impact of injection with the AMPA receptor antagonist CNQX. So I think it's pretty clear and pretty safe to assert that circulating epinephrine exerts a memory-enhancing effect in the presence of emotional arousal. And this effect seems to be mediated by a chain of communication from adrenergic receptors on the vagus nerve to the nucleus of the solitary tract, to the locus coeruleus, and ultimately triggering release of norepinephrine onto neurons of the basolateral amygdala. But there's also evidence from microdialysis studies that inhibitory avoidance increases extracellular norepinephrine content in the amygdala. And moreover, the latency to enter the zone where the shock was administered correlated with the amount of norepinephrine that was released into the amygdala. Okay, let's first make certain that everyone is up to speed on what a microdialysis experiment is all about, depicted in the images at the left side of this slide. In this paradigm, a probe with a semi-permeable membrane is inserted into a target site in the brain of the rat. The probe is then dialyzed with an artificial extracellular fluid, meaning that a clear solution with appropriate concentrations of ions is slowly flushed through the probe and then collected out the other side. As there are no neurotransmitters in the dialysate and the membrane is semi-permeable to neurotransmitters, they will flow by osmosis down their concentration gradient into the dialysate in proportion to the amount of neurotransmitter in the extracellular fluid surrounding the probe. So you can collect the dialysate, let's say every 10 minutes or so, and analyze the content of the dialysate to get an approximation of the amount of various neurotransmitters in the brain region you're studying. Now, if we look at the graph at the top right, we can see that the rats that underwent inhibitory avoidance conditioning had greater amounts of norepinephrine in the amygdala than did the rats that were not conditioned. And even more than rats that were not conditioned but actually got some equivalent shocks in their home cage. And in the bottom graph, the elevations in amygdaloid norepinephrine over time are plotted for individual rats, along with a latency to enter the shock zone in a 600-second or 5-minute test. Sorry, 10-minute test. The rats that did not enter the shock zone are given a score of 600. And you can see that these rats all basically had very high concentrations of norepinephrine in their basolateral amygdala, or in their amygdala in general, as measured with microdialysis. And then the rats that had shorter latencies to enter the shock zone tend to have lower release of norepinephrine in the amygdala. Now, I love this kind of experiment. Often we look at group means and just get the rough pictures of the events that are going on, as in the top graph. But a deeper delve into the individual differences that show up in the dependent measures, I think, can often be really revealing, as they are in this case. The conclusion is that the amount of modulating norepinephrine input from the brainstem to the amygdala is an important modulator of the strength of learning in the inhibitory avoidance task. Now, it has also been shown that microinjection of a low dose of norepinephrine right into the basolateral amygdala enhances learning and memory in the Morris water maze. This was not seen when a higher dose was administered and was blocked by the administration of the adrenergic antagonist, propranolol. And this experiment on the left side of the slide shows just how important the amygdaloid connection is. If you give a systemic injection of epinephrine, you get enhanced learning in the inhibitory avoidance test. Now, no surprise there. But the interesting part is that all you need to do to completely block that enhancement is to administer a microinjection of propranolol directly into the amygdala. So out of all the places where the circulating epinephrine could have its effects, it seems that the circuitry from vagus to nucleus of the solitary tract to locus coeruleus to basolateral amygdala is absolutely essential, because blocking that last link in the chain undoes the whole effect of a systemic injection. So, of course, we can relate all of these actions back to the mechanisms that are occurring at synapses. The sites of biochemical actions are going to invoke the players that we've addressed previously. So these cascades should seem familiar. A variety of experiments have shown us that norepinephrine binding to adrenoceptors in the basolateral amygdala activates adenylate cyclase. So elevated cyclic AMP binds to the regulatory subunits of protein kinase A, and increased protein kinase A phosphorylates ion channels and causes increased glutamate release from basolateral amygdaloid neurons. Then, on target neurons of the basolateral amygdala projections, increased glutamate release enhances mGlu-R activation to signal through G proteins to activate phospholipase C, and so on and so on. All the mechanisms that we've previously looked at for potentiation of neurons. The net result is that a variety of measures of hippocampal function are increased after inhibitory avoidance training. The experiment shown on the right measured one of these outcomes. When a very mild shock and minimal training was used in an inhibitory avoidance task, the animals did not develop very strong avoidance, as illustrated by the short short latencies on the graph. But when the adrenergic receptor agonist clenbuterol was injected into the basolateral amygdala, the latencies were significantly greater. And when the basolateral amygdala was injected with lidocaine, an anesthetic, the learning was eliminated, and the rats immediately entered the shock zone. Now, in the rats that were treated with clenbuterol during inhibitory avoidance training, you can also see that they have an elevated expression of ARC protein in the hippocampus. So the summary of all these experiments is simply that enhancement of learning by emotionally induced arousal appears to be mediated by the chain of processing that I outlined, from circulating epinephrine to vagus nerve to nucleus of the solitary tract to locus coeruleus to basolateral amygdala, and then out to the many sites to which amygdaloid afferents project. At those various sites, including the hippocampus and the caudate and other regions, depending upon what kind of task is being performed, we could see synaptic potentiation that is enhanced by the enhanced actions, the modulatory actions of the neurons of the basolateral amygdala. And I would be remiss if I didn't actually point out and show you that the locus coeruleus, this actually very tiny little pair of midline brainstem nuclei, provides noradrenergic innervation of structures all over the brain, contributing profoundly to maintenance of cortical arousal and readiness to learn. I believe there's a lot yet to learn about the importance of noradrenergic innervation of multiple different brain sites in mediating many different forms of learning. Okay, so let's look at an additional function of elevated epinephrine in the circulation. We've already seen the mechanism whereby circulating epinephrine gains access to the central nervous system through vagus signaling. But there's also a second route, and that is more indirectly through the release of stored glucose in the liver. Now, the liver is your principal store of glucose in the form of glycogen. There are two main signals that induce the liver to liberate glycogen into the circulation in the form of freely available glucose. One of those signals is the peptide hormone glucagon, which is released from the pancreas. The other is circulating epinephrine, which we've seen can be activated acutely by a challenging or stressful situation that activates the sympathetic branch of the autonomic nervous system. But, well, what does glucose have to do with enhanced learning and memory? Again, funny you should ask. Let's see. Of course, everyone should remember that the brain is only about 2% of the body mass, but it uses more than 20% of the energy in your body. And of course, that energy comes primarily in the form of glucose. The brain is highly privileged in that under normal circumstances, it uses glucose at all times, in contrast with the cells of your body that use glucose only when it is highly available, and they convert to other sources when glucose is not so abundant in the circulation. Neurons and glial cells in your brain break down glucose by engaging three processes known as glycolysis, Krebs citric acid cycle, also known as the tricarboxylic acid cycle, and the pentose phosphate pathway. It's very efficient in using these pathways to supply the energy needs of the cells. In fact, the brain, in contrast to the muscles, cannot store much energy. But like muscle cells, the neurons have greatly increased metabolic energy needs whenever they engage in increased activity. This includes learning and memory, as the genomic cascades, transcription and translation, and even post-translational processing all require energy. To say nothing of the increased load required by the sodium-potassium pump when the frequency of firing of action potentials increases. So wouldn't it be handy if there was a mechanism to couple energy supply to the neuronal demand in times when emotional stimuli are present and learning is occurring? And hey, this slide looks familiar. If you go back about 10 slides, you'll see a very similar slide. Except that in the previous slide, I showed you the impact of systemic norepinephrine injections on inhibitory learning. And of course, we saw that injection of norepinephrine at reasonable doses enhanced that inhibitory avoidance learning. So this is the same kind of story, but this time with injections of glucose. If moderate doses of glucose are injected into rats immediately after training, the latencies to enter the shock context are increased, demonstrating enhanced learning. Now, this effect doesn't occur when a high dose is injected, and similarly does not occur when a moderate dose is injected 1 hour after the training. So these data reveal that elevated glucose can actually enhance learning. Think about that the next time you want to take that ice cream break in the middle of a study session. So the finding that glucose can actually enhance learning and memory may actually have particular relevance for cognitive decline in aging. It's very common in the aged that there are deficits in management of glucose and availability of energy. So Morris and Gold trained rats in an inhibitory avoidance task under training conditions that were easily maintained over 7 days by the young rats. You can see that nearly every single one of the young rats did not enter the shock context in the 10-minute test 1 week after the training. Now, half the young rats were actually injected with glucose into the hippocampus, and half the rats were injected with artificial cerebrospinal fluid into the hippocampus at the time of training. Okay, but let's take a look at the aged rats under the same training conditions. You can see that the aged rats that were injected with the artificial CSF solution, the controls, readily venture into the shock context, demonstrating no retention of the inhibitory avoidance 1 week after the training. However, in those aged rats that received immediate post-training intrahippocampal glucose, this reversed the aging effect and allowed the aged rats to consolidate the memory, so they were just about as good as the young rats, staying out of the shock context for the duration of the test session in nearly each and every one of these aged rats. So the injection of glucose into the hippocampus appears to have reversed the age-associated memory impairment. Now, Morris and Gold also sought a biochemical marker to explain their finding. In this case, they used Western blots and phosphospecific Western blots to compare the ratio of phosphorylated Krebs to Krebs in the hippocampus of young and old rats trained in inhibitory avoidance. In the young rats, the ratio of Krebs to phosphorylated Krebs was elevated by training in the inhibitory avoidance task. And this elevated phospho-Krebs to Krebs ratio was further enhanced if the young rats received systemic injection of either epinephrine or glucose immediately after the training. And so this suggests that norepinephrine signaling and availability of glucose can both enhance the genomic signaling cascade initiated by the inhibitory avoidance training. In the aged rats, the effect of training alone was not observed. They had deficits in the expected training-related increases of phospho-Krebs to Krebs ratios, and so the ratios in the inhibitory avoidance trained rats were about the same as they were in the untrained elderly rats. However, injection of epinephrine or glucose normalized their training-induced elevations in the phospho-Krebs to Krebs ratio. So the epinephrine and glucose-treated rats had phospho-Krebs to Krebs ratios that resembled the ratios in the young rats. Morris and Gold concluded that arousing events may trigger increases in circulating epinephrine in aged animals that's roughly equivalent to the response seen in young animals. But impaired glucose response to the elevated epinephrine may also impair the learning capacity so that the aged animals experience a more rapid decline in the memory storage. Gold and Coral also provided an interesting model to summarize the interactions between epinephrine and glucose as it pertains to healthy young animals. In their model, an arousing event triggers an increase in autonomic arousal. So there's an increase in release of epinephrine into the circulation from the adrenal medulla. And this triggers an increase in glycolysis in the liver to liberate glucose into the circulation. Glucose, of course, enters the brain, is taken up by active neurons, and supports the energy demands to promote neurotransmitter release and increases in cell signaling and memory enhancement. On the other hand, a neutral stimulus fails to arouse the sympathetic nervous system, so it fails to activate the release of epinephrine, and from there, the rest of the steps required to enhance the memory incorporation are lost. The neutral event simply fails to endure in memory in the way that an emotionally charged event will. And of course, this fits with what psychologists have documented over many, many years, that emotionally charged or emotionally salient stimuli promote enhanced recognition and incorporation of memory, and neutral stimuli are not so likely to be incorporated. So let's check back with that modified diagram that I showed you at the beginning of this lecture. Except now, I've added another component. Now, if I don't quit adding new elements, I'm going to run out of room. But you can see that I've added an additional role for epinephrine, causing glycolysis from the liver to increase circulating glucose, and thereby supporting the neurons that are involved in memory consolidation for emotionally salient stimulus events. And if you look at the bottom of the diagram, you will see that there's one more connection that we have to make, and that is the connection of glucocorticoids. Mostly cortisol, if you happen to be a human, which I think most of us are, or mostly corticosterone, if you happen to be a rat. But these hormones are equivalent, and they do pretty much the same things in humans and rats. So let's see. Glucocorticoids, cortisol and corticosterone, are lipophilic steroid hormones, synthesized mostly by cells of the adrenal cortex in response to signals from the hypothalamic pituitary inputs. You can see the gross anatomy of the system in the figure at the right. And then let's take a closer look at the brain and pituitary mechanism in the figure at the left. In fact, emotionally evocative and stressful stimuli activate limbic inputs to the parvocellular neurons of the paraventricular nucleus of the hypothalamus. These neurosecretory parvocellular neurons release the neurohormones corticotropin-releasing hormone, or CRH, and arginine vasopressin, or AVP, into the hypophyseal portal system at the plexus of capillaries known as the median eminence. The portal system then conveys these neurohormones to the anterior pituitary to induce corticotrope cells to release adrenocorticotropic hormone, or ACTH, into the general circulation. The ACTH circulates to adrenocortical cells and induces those cells to release the glucocorticoid hormones. Now, I expect that was a quick refresher on the hypothalamic pituitary adrenal axis, or HPA axis, for most of you, but I hope it reminds everyone of the connection between emotional stimuli and increases in circulating glucocorticoids. Of course, those glucocorticoids are steroid hormones, so they are lipophilic and can enter all cells of the body. Now, in the brain and pituitary, there's a high abundance of cytosolic glucocorticoid receptors, commonly called GRs, and binding of glucocorticoid hormones to these receptors provides a negative feedback mechanism to self-regulate the HPA axis. Binding to GRs also causes a broad variety of cellular responses in neurons, including lots of cellular signaling and activation of particular genomic cascades. But let's connect glucocorticoid actions with learning. Again, we're looking at experiments on inhibitory avoidance learning. In this case, rats had surgical lesions of the central amygdala or basolateral amygdala, or they had sham surgeries with no damage to the brain. The rats were trained in inhibitory avoidance, and systemic injections of the glucocorticoid receptor agonist dexamethasone were administered immediately after the inhibitory avoidance training. The rats that had sham surgeries and were injected with dexamethasone exhibited greatly enhanced latency to enter the shock context when tested 48 hours after the training. Accordingly, the DEX treatment appears to have enhanced the retention of the inhibitory avoidance. In the rats with lesions of the basolateral amygdala, the dexamethasone treatment was ineffective, and these rats had very short latencies to enter the shock context. And in the rats with lesions of the central nucleus of the amygdala, the dexamethasone treatment was still able to enhance the inhibitory avoidance. These data suggest that the glucocorticoid receptor-mediated enhancement of inhibitory avoidance resulted from actions of the dexamethasone in the basolateral amygdala. In an additional experiment, Gina Kirarte, another name that I'm certainly butchering, trained rats in inhibitory avoidance and then immediately injected the glucocorticoid receptor agonist RU28362 into the basolateral amygdala. The photomicrograph in the middle of the page shows an injector track into the basolateral amygdala. The graph at the left shows that moderate doses of RU28362, the glucocorticoid receptor agonist, into the basolateral amygdala enhance retention of the inhibitory avoidance measured 48 hours after the training. Then in the figure at the right, the rats were injected systemically with dexamethasone, the other glucocorticoid receptor agonist that we've been looking at. And then they were injected into the basolateral amygdala with the beta-adrenergic antagonist propranolol. This experiment shows again that glucocorticoid actions enhance inhibitory avoidance learning, and that this effect is actually dependent upon norepinephrine actions in the basolateral amygdala. When we block the noradrenergic function in the basolateral amygdala, we eliminate the enhancing effect from a systemic injection of dexamethasone. So once again, we see a connection with noradrenergic neurotransmission. Phew. Okay, let's sum this up. And once again, I'm going to use my enhanced diagram to see if we can make this all make sense. And certainly it does. We've seen evidence that the basolateral amygdala is the linchpin for modulation of memory consolidation, particularly for emotionally salient events. Neutral events fail to initiate the adrenergic and glucocorticoid cascades that result in memory enhancement. But emotionally salient events activate all these cascades to invoke signaling cascades that activate the sympathetic nervous system and HPA axis. These activities cause peripheral release of epinephrine and glucocorticoids from the adrenal medulla and cortex, respectively. The circulating epinephrine activates a chain of neural signaling involving the vagus nerve, nucleus of the solitary tract, and locus coeruleus that eventually releases norepinephrine at the basolateral amygdala and many other sites throughout the brain. Peripheral epinephrine also increases release of glucose from the liver that supports neuronal encoding of memories at the basolateral amygdala and certainly other brain regions. And release of glucocorticoids also stimulates the basolateral amygdala and other brain regions to interact with learning and memory mechanisms. So it really does appear that the basolateral amygdala integrates a whole lot of different information from noradrenergic innervation, glucocorticoid action, glucose stimulation, to produce a profile of functioning that represents that there is arousal, that there is emotional salience to promote the incorporation of the memory. So, should we all go out and take some amphetamine? Or perhaps a dose of sweet treats, or maybe watch a scary movie to get some emotional arousal in order to facilitate our studying and our learning and memory performance? Well, of course, the answer is no. We did see some implications for interactions between epinephrine and glucose metabolism in the elderly subjects, and those were rats actually. And we know that the doses of stimulants that promote cognitive function are way less than the ones that humans take, and that their contribution to cognitive enhancement is minimal at best. So we've used these manipulations to map out the mechanisms that underlie enhanced retention of emotionally salient memories, but that doesn't mean that these mechanisms can necessarily be enhanced in normal learning in a human. In fact, in a healthy young brain, the best prescription for enhanced learning is time-limited bouts of studying, as we saw previously, and plenty of sleep. And I think I'm going to add some information on the important role of sleep in an upcoming chapter.